Imagine you stand in quiet Cambridge laboratory, April 25th, 1953. Late evening. Only sounds, soft hum of cooling ultracentrifuge, occasional pipette click from adjacent bench, distant rain falling on cobblestone courtyard outside Cavendish Laboratory. On wooden desk sits Photo 51, Rosalind Franklin's X-ray crystallography image showing DNA's B-form helix structure. Clear X-shaped pattern emerges from silver halide crystals exposed to DNA fibres arranged parallel, diffraction pattern revealing hidden molecular geometry. Nearby, notebook pages filled with precise measurements written in pencil. 3.4 angstroms per base pair, turn, 34 angstroms, helical repeat distance, diameter approximately 20 angstroms. James Watson sketches double helix model from these measurements, cardboard cutouts representing bases. Francis Crick calculates bond angles on slide rule, checking whether adenine-thymine and guanine-cytosine pairs fit sterically within helix diameter. Tobacco smoke drifts lazily from ashtray. Laboratory windows fog slightly from humid spring air mixing with warm interior. Patient geometry of life's blueprint slowly reveals itself through night's quiet concentration. Good evening. Welcome back. New listeners, welcome. Tonight, perfect genetic rest awaits. Find comfortable position wherever you listen. Dim lights completely, if possible. Adjust pillow beneath neck. Perhaps soft background hum mimicking PCR thermal cycler, rhythmic temperature changes. Let breathing slow to match DNA replication rhythm, steady and patient. Perfectly safe drifting among nucleotides and laboratory benches. Tonight, slowly explore complete DNA history. Mendel's monastery pea garden through Watson-Crick double helix. Discovery to CRISPR gene editing revolution. Double helix. Watch inheritance patterns emerge mathematically from simple counting, double helix structure, crystallize from X-ray diffraction shadows, genetic code deciphered letter by letter through biochemical detective work, gene editing molecular scissors cutting precisely at targeted sequences. Long patient journey through laboratories worldwide, told with maximum atmospheric detail and sensory immersion. Perfect, gentle drifting through base pairs and breeding experiments. If genetic stories help restful sleep, consider like or subscribe. Curious what time zone you're listening from? Comment your city or location. No pressure whatsoever, just relax into story. Now you find yourself transported back through time and space, appearing in different form in monastery garden in Brno, Austrian Empire, year 1856. You stand among carefully tended pea plants growing in neat rows within Augustinian monastery grounds. Friar Gregor Mendel, 34 years old, physics and mathematics teacher at local gymnasium, walks slowly between plant rows carrying notebook and pencil. Late spring morning, cool but sunny, bees humming among blossoms. Garden soil dark and moist from overnight rain, earthworm castings visible between pea plant bases. Green vine tendrils curl around vertical strings tied to wooden stakes, plants climbing upward seeking sunlight. Mendel pauses before plants bearing flowers, examining closely. He carries small brush made from rabbit hair, fine bristles perfect for transferring pollen between flowers without damaging delicate reproductive structures. Morning dew still clings to leaves, water droplets refracting sunlight into tiny rainbows. Carefully opens flower petals, revealing reproductive structures. Stamens bearing pollen-laden anthers, pistil with sticky stigma ready receiving pollen. Uses magnifying lens checking anthers' maturity, looking for yellow pollen grains powdery and dry rather than sticky and immature. When satisfied with readiness, gently brushes anthers, collecting pollen grains, adhering to soft bristles, then carefully applies accumulated pollen to stigma of different flower, transferring genetic material between plants deliberately rather than allowing bees random pollination dictated by flower visiting preferences and flight patterns. After controlled pollination complete, wraps flower in paper bag, preventing accidental cross-pollination from insects attracted to flower colours and scents, records cross in notebook with careful handwriting noting parent plant's characteristics observed across previous generations. This meticulous process continues daily throughout growing season from May through August. Mendel focuses on 7 distinct traits, showing clear either-or differences without intermediate forms. Seed shape round versus wrinkled when dried, seed colour yellow versus green, visible through translucent seed coat, flower colour purple versus white visible along corolla, pod shape smooth versus constricted, showing indentations between developing seeds, pod colour, green versus yellow when immature, flower position, axial growing from leaf nodes versus terminal clustering, at stem tip, stem length tall, exceeding 6 feet versus short, remaining under 18 inches. Each trait inherited independently as discrete unit rather than blending, like paints mixing purple and white flowers, never producing pale purple offspring. This insight revolutionary, though Mendel doesn't yet know its far-reaching significance for understanding heredity. You watch summer pass slowly through monastery garden. Days blend into weeks marked only by plant growth stages, weeks into months distinguished by shifting angle of sunlight across garden rows. Mendel visits garden daily, regardless of weather conditions, sometimes multiple times when pollination work demands, checking plants during early morning coolness before heat arrives, making crosses during midday when flowers fully open, recording observations during evening hours before Vespers prayers. Garden becomes second home, familiar as monastery chapel where he attends daily services. He knows individual plants by their position in specific rows, remembers lineage of each plant tracing back multiple generations, tracks inheritance patterns across pedigrees with mathematical precision unusual for biological studies in this era dominated by qualitative natural history descriptions. Most naturalists describe organisms' appearances, habitats, behaviours qualitatively using prose. Mendel counts and calculates, applying physics approach learned during university studies to living things, quantifying ratios and testing hypotheses through statistical analysis decades before such methods become standard practice in biology. First-generation seeds develop within pods as summer progresses week by week. Pods swell gradually from small green nubbins to full-sized structures, green and taut, photosynthesizing sugars actively through chlorophyll-rich pod walls, while simultaneously filling developing seeds within. Inside each pod, seeds grow from tiny ovules into plump, mature seeds packed with starch reserves. By late summer, around August, pods dry progressively to brown, papery texture as plants senesce and nutrients mobilize into seeds, pods rattling audibly when shaken, indicating seeds dried sufficiently for harvesting. Mendel harvests carefully, opening each pod individually using fingers to shell peas, labels each pod's seeds by parentage information, stores in small paper envelopes folded and sealed with paste, marked with precise breeding information written in ink specifying both maternal and paternal parent plant characteristics. Each envelope represents distinct genetic experiment, hidden inheritance rules, waiting discovery through patient systematic counting across generations. Autumn arrives gradually as September days grow shorter and cooler. Garden leaves turn yellow, then brown, Temperature drops steadily, forcing outdoor work to conclude before frost arrives. But Mendel's scientific work continues indoors throughout autumn and winter months. He examines harvested seeds under oil lamplight in his monastery room during long evening hours after teaching duties complete, categorizing meticulously by observable phenotypic traits. Round seeds feel smooth to touch like glass beads. Wrinkled seeds have deeply irregular corrugated texture resembling dried raisins, difference immediately apparent when rolling seeds between fingers. Yellow seeds bright and cheerful like sunshine, ranging from pale cream to deep golden yellow, green seeds darker, more muted olive tones sometimes tinged with bluish cast. He counts every single seed exhaustively, recording numbers in ruled ledger books with careful penmanship, columns of figures marching down pages. 5,474 round seeds, 1,850 wrinkled seeds from specific round wrinkled F1 hybrid cross self-pollinated producing F2 generation. Ratio approximately 2.96 to 1, remarkably close to theoretically perfect 3 to 1 ratio expected if trait controlled by single gene with complete dominance. Similar mathematically precise ratios emerge from analyzing other traits independently. Yellow-green seed crosses yield 6,022 yellow versus 2,001 green. F2 seeds, ratio 3.01 to 1. Purple-white flower crosses produce 705 purple versus 224 white flowers, ratio 3.15 to 1. Pod colour, green versus yellow, shows 428 green versus 152 yellow, ratio 2.82 to 1. Seed coat texture smooth versus constricted yields 882 smooth versus 299 constricted, ratio 2.95 to 1. Every trait tested shows same fundamental 3 to 1 pattern within statistical variation expected from finite sample sizes. Pattern consistent across thousands of individual plants grown, counted, analysed over 8 years continuous experimentation. Something fundamentally mathematical underlying inheritance, discrete hereditary factors segregating predictably during reproduction according to probability laws governing random independent events. Mendel hypothesizes each trait controlled by paired factors, what we now call alleles, one factor inherited from each parent through male pollen and female ovules, some factors dominant, expressing their effects even when paired with different recessive factor, other factors recessive, only producing visible effects when both inherited factors identical, making homozygous recessive genotype, heterozygotes phenotypically indistinguishable from homozygous dominance. You watch years cycle through similar patterns, each spring planting new generation seeds, carefully maintaining pure breeding lines through controlled self-pollination, also making specific test crosses exploring inheritance patterns, F3 testing, whether F2 plants showing dominant traits breed true or segregate indicating heterozygosity. Each summer monitoring plant development, measuring heights, counting flower positions, noting seed pod characteristics before harvest. Each autumn harvesting seeds, sorting by traits, counting exhaustively, entering numbers in ledgers, calculating ratios, checking predictions against mathematical models derived from probability theory. Each winter analysing accumulated data, recognizing patterns, formulating hypotheses, designing next-generation experiments, testing predictions. Hypnotic rhythm of agricultural genetics, patient systematic accumulation of evidence through countless individual observations repeated across years until patterns unmistakable, laws of inheritance crystallizing from numerical data. Winter 1857. Garden sleeps under snow. Mendel works indoors, planting spring seeds in greenhouse pots, planning next-generation experiments. He needs testing second generation, seeing if patterns hold. If hypothesis correct, self-fertilizing first-generation hybrids should produce 3:1 ratios in second generation, but then third generation should behave interestingly. Plants showing dominant traits should include both pure breeding and hybrid types in 1:2 ratio, while plants showing recessive traits should breed true always. Spring 1858. Second generation plants grow. Summer brings flowers. Mendel allows self-pollination this round, simply watching and counting. Autumn harvesting confirms predictions. Third generation planted 1859. Patterns hold beautifully. Numbers align with mathematical model, assuming independent segregation of paired factors during gamete formation. 8 years of experiments, 28,000 plants grown, counted, analysed. You watch seasons cycle repeatedly, same routine recurring with slight variations. Each spring planting seeds carefully, maintaining plant varieties separately, making specific crosses. Each summer monitoring growth, occasionally hand-pollinating, mostly allowing self-fertilization. Each autumn harvest, counting seeds, recording ratios, checking predictions. Each winter analysing data, calculating chi-square goodness of fit before such statistical tests formally exist, planning next experiments. Hypnotic rhythm of agricultural genetics, patient accumulation of evidence through countless individual observations. By 1865, Mendel presents findings to Natural Science Society in Brno across 2 evening lectures, February and March. Audience small, perhaps 40 members, mostly local naturalists, teachers, clergy interested in natural history. Mendel explains crosses, shows numerical data, proposes mathematical laws, law of segregation stating paired factors separate during gamete formation, with offspring inheriting one factor from each parent randomly. Law of independent assortment stating different traits inherited independently without influencing each other. Presentations received politely, but without enthusiasm. Members don't grasp significance, revolutionary implications lost on audience, lacking mathematical sophistication or understanding of hereditary mechanisms. Mendel publishes results 1866 in Local Science Society Proceedings, paper titled Experiments on Plant Hybridization. 40 reprints printed, mailed to prominent botanists and institutions across Europe. Response: silence. Paper largely ignored, occasionally cited but misunderstood, significance unrecognized for 34 years. Mendel continues teaching, becomes abbot of monastery 1868. Administrative duties consume time previously devoted to research. Dies 1884, having never witnessed scientific community embrace his discoveries. Work rediscovered independently by 3 botanists in 1900, Hugo de Vries in Netherlands, Karl Correns in Germany, Erich von Tschermak in Austria. Each rediscovers Mendel's laws while conducting own hybridization experiments. Each searches literature, finding Mendel had discovered same patterns 4 decades earlier. You now drift forward through time several decades, finding yourself in different laboratory, different continent. Columbia University, New York City, year 1910. You stand in small room designated fly room, officially Schermerhorn Hall, room 613, walls lined with shelves holding hundreds of milk bottles containing fruit flies, Drosophila melanogaster. Thomas Hunt Morgan, 44 years old, experimental embryologist recently converted to genetics research examines flies under binocular microscope wearing white lab coat. Several students work alongside Alfred Sturtevant, Calvin Bridges, Herman Muller, future genetics pioneers, currently graduate students cramming into close quarters. Laboratory smells distinctly of fermenting bananas and yeast mixed with ether's sharp sweet odour. Fly culture medium providing food for larvae, developing within bottles, crowding every available shelf surface. Each bottle contains mashed overripe banana mixed with agar powder and yeast cake. Consistency like thick pudding, providing carbohydrates, proteins, vitamins supporting rapid fly development from egg through 3 larval stages, pupation, to adult emergence. Cotton plugs stuffed firmly into bottle openings prevent fly escape while allowing air exchange necessary for aerobic respiration. Adult flies cluster on bottle walls and cotton surfaces, red eyes gleaming like tiny rubies under afternoon sunlight streaming through laboratory windows, small tan-coloured bodies roughly 2 millimeters long, wings folded delicately across backs when resting. Reproduction extraordinarily rapid compared to pea plants, generation time approximately 10 days under warm conditions maintained by steam radiators heating building, single female laying hundreds of eggs during reproductive lifetime, allowing multiple generations studied within months rather than Mendel's years-long pea breeding cycles spanning nearly a decade before patterns emerged clearly. This particular morning, spring 1910, Morgan notices unusual fly in one bottle while examining cultures during routine daily inspection. Most flies possess brilliant red compound eyes composed of roughly 800 individual ommatidia giving mosaic vision, but this lone male possesses stark white eyes completely lacking red pigmentation, startling deviation from normal wild-type appearance. Mutation appeared spontaneously in laboratory culture maintained for several months since capturing founding flies from fruit rotting in Market District. First recorded instance of visible mutation in Morgan's fly stocks, though he suspects many invisible biochemical mutations also occurring constantly. He immediately isolates white-eyed male carefully using ether anesthesia, begins breeding experiments testing inheritance pattern following Mendelian principles, crosses white-eyed male with normal red-eyed virgin females collected before any mating occurs, preventing contamination with unknown sperm from previous matings, carefully transfers virgins using ether and fine brush to fresh culture, bottle prepared with fresh banana medium alongside white-eyed male, seals with cotton plug, labels bottle with cross information and date using wax pencil on glass. You watch Morgan's anesthesia technique repeated hundreds of times daily across laboratory. He prepares etherizing apparatus, cotton wool soaked liberally with ethyl ether, placed in small glass chamber called etherizer, custom built by glassblower. Tips culture bottle, containing flies over etherizer opening, taps bottle gently against palm, causing flies tumbling onto ether-soaked cotton surface, where they quickly become immobilized by anesthetic fumes after several seconds exposure, causing reversible narcosis through disrupting neural membrane potentials. Morgan then carefully pours anesthetized flies onto white porcelain examination plate with shallow depression holding flies during sorting. Works quickly under dissecting microscope, illuminated by daylight reflected from adjustable mirror, before flies awaken after several minutes ether exposure, separating males from females based on body size, with females larger, and sex comb structures on male forelegs, plus differences in genital structures visible at posterior abdominal end under magnification, counts eye colour variants noting numbers in laboratory notebook, virgin females younger than 8 hours post. Eclosion transferred immediately to fresh culture bottles, with specific males preventing uncontrolled mating, complicating genetic analysis. First-generation offspring from white-eye male crossed with red-eye female emerge after 12 days egg to adult development. All offspring show red eyes, both males and females. White-eye trait behaves recessively like Mendel's factors. But second generation reveals surprising pattern, approximately 3 to 1 red to white ratio as expected from Mendelian genetics, but white-eyed flies exclusively male. No white-eyed females appear among thousands of offspring examined. This sex linkage strange, suggesting eye colour factor located on same chromosome determining sex. Morgan hypothesizes factors, now called genes, physically located on chromosomes visible under microscope during cell division. Sutton and Bovary proposed this correlation 1902 to 1903, suggesting Mendel's abstract factors corresponded to physical structures within cells, but lacked direct evidence. Morgan's white eye mutation provides crucial link. If eye colour gene resides on X chromosome, sex-specific inheritance pattern explained perfectly. Females possess 2 X chromosomes, males 1X and 1Y, therefore recessive mutations on X chromosome would affect males lacking second X copy, masking recessive allele. You watch Flyroom evolve into remarkable genetic factory. More mutations discovered monthly, some affecting eye colour, vermilion, sepia, brown, scarlet, others affecting wing shape, body colour, bristle patterns. Students map genes to chromosomes through breeding analysis, calculating recombination frequencies between linked genes, constructing first genetic maps showing gene positions arranged linearly along chromosomes. Sturtevant, undergraduate student, works through night in January 1913, constructing first chromosome map instead of studying for examination next day, creating spatial representation of genetic linkage relationships, founding principle of gene mapping still used today. Laboratory atmosphere intense but collegial. Students discuss crosses constantly, debate interpretations, propose new experiments. Morgan encourages independent thinking while maintaining rigorous standards for data quality. Every claim requires solid evidence from large sample sizes, ambiguous results repeated until clarity emerges, false leads acknowledged and abandoned. Scientific method practiced seriously, despite cramped quarters and limited funding. Space so restricted that desks pushed against walls barely allow passage between, yet productivity remarkable. Group publishes landmark book The Mechanism of Mendelian Heredity in 1915, firmly establishing chromosome theory of inheritance through accumulated experimental evidence from thousands of fly crosses. Cytological examination confirms breeding predictions. Careful microscopy of fly salivary gland chromosomes. Giant polytene chromosomes produced through repeated DNA replication without cell division, resulting in parallel chromatids visible as banded patterns under microscope, allows correlating visible chromosome abnormalities with genetic mapping data. Deletions, duplications, inversions, translocations visible as altered banding patterns correspond to genetic linkage map changes, providing direct visual proof that genes reside at specific chromosomal positions. Abstract genetic factors become concrete physical entities observable under microscope. But what exactly are genes chemically? This question remains mysterious. Chromosomes contain both protein and nucleic acid, both candidates for genetic material. Most biochemists assume proteins more likely, offering greater chemical complexity through 20 amino acid building blocks versus nucleic acid's 4 bases. You drift backward now temporally, visiting earlier moments in genetics history, addressing this chemical mystery. You find yourself standing in University of Tübingen Laboratory, Germany, year 1869. Friedrich Miescher, 25-year-old Swiss physician, recently completed medical degree, works at bench extracting chemical components from white blood cells obtained from surgical bandages discarded by local hospital. Pus collected from bandages provides abundant white blood cells, leukocytes, for biochemical analysis. Miescher washes bandages with sodium sulfate solution, allowing pus cells to settle, then treats cells with weak acid, extracting lipids and proteins. Laboratory kept intentionally cold during these winter months, utilizing natural outdoor cold for preserving biological samples from bacterial and enzymatic degradation before modern refrigeration technology exists beyond ice harvesting. Miescher wears heavy wool coat and scarf while working during December and January, breath occasionally visible in chilly laboratory air dropping to perhaps 10 degrees Celsius. Fingers sometimes numb, requiring warming periodically near coal stove in adjacent room. He pipettes carefully using glass pipettes with rubber bulbs, watching reactions proceed in glass test tubes held against window light streaming through laboratory windows, observing colour changes and precipitation patterns indicating chemical transformations occurring. After initial acidic extraction, successfully removes most cellular proteins through precipitation at low pH, where proteins lose solubility. Subsequent alkaline treatment using dilute sodium hydroxide solution raises pH, dissolving remaining cellular material, including nuclein, which remains soluble at alkaline pH, leaving peculiar white fibrous substance when solution acidified again, causing nuclein precipitating out of solution as visible white threads that Miescher can wind onto glass stirring rod-like cotton thread, being spooled. Material exhibiting unusual physico-chemical properties unlike any known biological substance. Substance unusual in multiple respects. Extraordinarily high phosphorus content detected through chemical colour tests involving molybdate reagent producing yellow precipitate, proportional to phosphate concentration, soluble readily in alkaline solutions, but quantitatively precipitates upon acidification, indicating amphoteric behaviour. Highly resistant to protein-degrading enzymes, including pepsin, trypsin, and chymotrypsin. Tested systematically yet degraded by unknown enzymes in pancreatic extracts, later identified as deoxyribonucleases. He names mysterious substance nuclein. Believing correctly, it originates primarily from cell nuclei, based on cytological observations showing nuclei rich in this phosphorus-containing acidic material, staining intensely with basic dyes. Further purification reveals nuclein contains organic bases, phosphoric acid, and sugar molecules. Miescher publishes findings 1871 describing novel acidic substance from cell nuclei, distinct from all known biological molecules. Scientific community takes little notice. Nuclein seems metabolic waste product or structural component, unlikely to possess functional significance given apparent chemical simplicity compared to proteins' versatility. Miescher himself hypothesizes proteins carry hereditary information while nuclein provides structural scaffolding, intuition shared by most biochemists for next 7 decades. Yet evidence gradually accumulates favouring nucleic acids, renamed nucleic acids by Richard Altman in 1889, distinguishing deoxyribonucleic acid from ribonucleic acid by presence of ribose versus deoxyribose sugar. Phoebus Levine at Rockefeller Institute characterizes nucleic acid structure 1919 to 1930, identifying 4 nitrogenous bases, adenine, guanine, cytosine, thymine, attached to sugar phosphate backbone, proposing bases arranged as repeating tetranucleotide units. This tetranucleotide hypothesis suggests overly simple regular structure incapable of encoding complex hereditary information, further supporting protein genetic material hypothesis. You drift forward to 1928, finding yourself in laboratory at British Ministry of Health, London. Frederick Griffith, medical officer researching pneumonia bacteria, conducts experiments with 2 strains of Streptococcus pneumonia. One strain, smooth colonies on agar plates due to polysaccharide capsule protecting bacteria from immune system, causes fatal infections in mice. Other strain, rough colonies lacking capsule, harmless to mice since immune system readily destroys unprotected bacteria. Griffith injects mice with various bacterial preparations, testing whether immunity develops. Critical experiment involves injecting mice with mixture of heat-killed smooth bacteria plus live rough bacteria. Heat killing destroys smooth bacteria completely. They cannot grow when cultured alone on plates. Rough bacteria alone, harmless. But mixture proves deadly. Mice die, blood cultures grow smooth bacteria recovered from dead mice. Somehow rough bacteria acquired ability to produce protective capsule, transformed from harmless to virulent type. Transformation persists through subsequent bacterial generations, suggesting heritable change occurred rather than temporary modification. You watch Griffith's meticulous experimental technique developed through years working with pathogenic bacteria. He prepares bacterial cultures on blood agar plates containing 5% defibrinated sheep, red blood cells mixed with nutrient, agar providing iron and other growth factors, pours molten agar into sterile glass petri dishes, allowing solidification, incubates inoculated plates overnight at 37 degrees Celsius in warm thermostatically controlled incubator resembling insulated cupboard with gas flame heating and mercury thermometer regulator. Next morning examines plates, noting colony appearances. Smooth type colonies appear glistening and mucoid from polysaccharide capsule secretion, forming characteristic greenish alpha hemolytic zones where bacterial hydrogen peroxide partially lyses surrounding red blood cells without complete clearing. Rough type colonies appear dull matte finish surfaces, dry looking due to absence of capsule, distinctly smaller diameter, typically 1 to 2 millimeters, versus smooth colonies reaching 4 to 5 millimeters. Griffith picks individual well-isolated colonies using platinum wire loop, sterilized first in Bunsen burner flame until glowing red hot, then cooled briefly in air, transfers single colony to liquid Todd-Hewitt broth medium containing peptones, yeast extract and glucose, grows overnight at body temperature, shaking culture flask, gently providing aeration for optimal bacterial respiration. Next morning measures bacterial density using simple turbidity observation, comparing culture cloudiness against McFarland nephelometer standards, dilutes cultures to standard concentrations for injection using sterile saline solution, prepares injections using glass syringe fitted with fine needle sterilized by autoclaving under steam pressure. Mouse experiments require exceptionally careful sterile handling, preventing accidental contamination and ensuring reproducible results. Griffith uses white laboratory mice weighing 20 to 25 grams, approximately 8 weeks old, housed in wire cages with food pellets and water bottles changed daily. Housed in He injects bacterial suspensions subcutaneously under skin of abdominal wall using volume typically 1/2 milliliter, containing known number of bacterial cells determined by plate counting method. Observes mice carefully twice daily for clinical signs of pneumococcal infection. Initial lethargy progressing to severely ruffled fur, laboured respiration with rapid shallow breathing, hunched posture, reduced activity culminating in death, typically occurring 24 to 72 hours post-infection for virulent smooth bacteria. Sick mice approaching moribund state sacrificed humanely using chloroform, anesthesia, overdose or cervical dislocation preventing suffering, blood samples collected aseptically from heart using syringe and needle sterilised immediately before use. Blood plated immediately onto fresh blood, agar plates incubated overnight, bacterial types present in blood identified next day based on colony morphology. Transformation phenomenon reproducible across dozens of independent experiments. Mixed injection of heat-killed smooth pneumococcus bacteria plus live rough bacteria consistently yields live smooth bacteria recovered from blood of dead mice, exhibiting smooth colony morphology and virulence properties, while all control experiments show expected results. Heat-killed smooth bacteria alone causing no infection. Mice surviving healthy, live rough bacteria alone causing no disease, mice surviving only mixture of heat-killed smooth plus live rough producing fatal transformed infection. Griffith publishes findings 1928, proposes transformation represents heritable change, but does not speculate regarding biochemical mechanism. Scientific community intrigued but puzzled. What substance from dead smooth bacteria transforms living rough bacteria? Attention focuses on identifying transforming principle responsible for genetic change. You drift across Atlantic Ocean to Rockefeller Institute Hospital, New York City, 1944. You stand in laboratory of Oswald Avery, 67 years old, Canadian-American physician and molecular biologist who spent 16 years since Griffith's discovery meticulously identifying transforming principles' chemical nature. He works with two colleagues, Colin MacLeod and Macklin McCarty, isolating and purifying transforming activity from pneumococcus bacteria, testing which molecular class, protein, lipid, carbohydrate, or nucleic acid, carries genetic information. You watch patient purification procedure unfold across many months of careful, meticulous work consuming countless hours in laboratory. Starting material consists of huge cultures of smooth-type pneumococcus bacteria grown in hundreds of glass Erlenmeyer flasks, each containing 1 litre rich medium composed of beef heart infusion supplemented with glucose and amino acids, incubated overnight at 37 degrees Celsius in temperature-controlled incubator, producing turbid bacterial suspensions cloudy with billions of cells per milliliter. Bacteria harvested next morning by centrifugation, spun at high speed 3,000 revolutions per minute for 30 minutes in angle. Head centrifuge producing tremendous centrifugal force, separating dense bacterial cells from liquid growth medium, pouring off supernatant liquid, leaving thick paste-like bacterial pellet adhering to tube bottom. Cells lysed using sodium deoxycholate detergent, bile salt disrupting bacterial membranes and cell walls through detergent action on lipid bilayers, rupturing cells and releasing entire cellular contents, including chromosomal DNA mixed with proteins, ribosomes, membranes, metabolites, all dissolved together in crude lysate, resembling thick viscous syrup. Crude lysate subjected to sequential purification steps, systematically removing different molecular classes through selective precipitation and enzymatic digestion. First stage removes proteins using chloroform, alcohol extraction, adding equal volume of chloroform isoamyl alcohol mixture to aqueous lysate in large separatory funnel, shaking vigorously for several minutes, creating emulsion of aqueous and organic phases, allowing phases to separate slowly over 30 minutes, yielding 3 distinct layers. Upper aqueous phase containing nucleic acids, middle whitish interface layer containing denatured proteins precipitated at phase boundary, lower clear organic phase containing extracted lipids. Carefully drain and discard protein-containing interface layer and organic phase. Retain aqueous phase containing transforming activity. Throughout purification, small aliquots of material tested repeatedly by adding extracts to recipient rough pneumococcus bacteria growing on blood, agar plates in separate sterile room, counting smooth colony transformants appearing after overnight incubation at body temperature, measuring transformation frequency as smooth colonies per 100,000 recipient cells plated, monitoring activity, retention through purification, confirming genetic activity not lost during chemical treatments. After removing bulk proteins through organic extraction, additional proteins eliminated using enzymatic digestion. Crystalline trypsin enzyme added to partially purified extract, incubated at 37 degrees for 1 hour, allowing protease enzyme cleaving peptide bonds between amino acids, hydrolyzing proteins into small peptides and amino acids, then heat treated to inactivate trypsin enzyme, preventing further unwanted proteolysis. Chymotrypsin added similarly, different protease enzyme with different specificity, ensuring complete protein degradation. Ribonuclease enzyme added digesting any contaminating RNA molecules present from bacterial ribosomes. After each enzymatic treatment, transformation activity assayed, confirming retention, chemical tests performed, measuring protein content through Biuret reaction, turning solution purple in proportion to peptide bond concentration, measuring nucleic acid content through diphenylamine reaction, producing blue color proportional to deoxyribose sugar content, monitoring purification progress objectively. After removing proteins, carbohydrates, lipids, activity persists. Chemical tests on purified material reveal high phosphorus content characteristic of nucleic acids, low sulfur content inconsistent with proteins. UV absorption spectrum shows peak at 260 nanometers, typical of nucleic acids rather than proteins' 280 nanometer peak. Enzymatic digestion experiments provide crucial evidence. Protease enzymes degrading proteins don't affect transforming activity. Lipase enzymes don't affect it. Carbohydrate cleaving enzymes don't affect it, but ribonuclease destroying RNA doesn't affect it either. Finally, newly discovered deoxyribonuclease enzyme, specifically degrading DNA, completely destroys transforming activity. Conclusion: inescapable, DNA carries genetic information. Avery publishes results 1944 in Journal of Experimental Medicine. Paper cautiously worded, acknowledging evidence points toward DNA, but noting possible contaminating proteins might account for activity. Many biochemists remain skeptical. DNA still seems too simple chemically to encode complex genetic information. Tetranucleotide hypothesis, suggesting monotonous regular structure, persists in many minds. Erwin Chargaff's studies, 1947 to 1952, disproving tetranucleotide hypothesis, show base composition varies between species. Ratios of adenine, thymine, guanine cytosine, different in bacteria versus mammals versus plants, suggesting DNA structure irregular enough to encode information. Chargaff also discovers Chargaff's rules. Amount of adenine equals thymine, amount of guanine equals cytosine. Puzzling observation lacking explanation until DNA structure solved. You drift now, through time, approaching culmination of DNA's chemical mystery. Alfred Hershey and Martha Chase perform elegant experiment 1952 using bacteriophage viruses infecting bacteria, radioactively labelling either phage, protein coats with sulfur 35, or phage DNA with phosphorus 32, showing only labelled DNA enters bacteria during infection while protein coats remain outside, definitively proving DNA carries genetic information from virus to bacterium, not protein. After this final demonstration, scientific consensus shifts. DNA is genetic material, but central structural question remains unsolved. How does DNA encode vast amounts of genetic information required specifying complex organism development? What precise three-dimensional molecular configuration allows DNA simultaneously storing genetic instructions while being accurately copied during each cell division, ensuring hereditary continuity across generations? How do four simple chemical nucleotide bases, adenine, guanine, cytosine, thymine, arranged along sugar phosphate backbone, create language capable encoding instructions, building complex proteins, regulating development, determining inherited characteristics? This brings us back full circle to Cambridge Laboratory, April 1953, quiet evening laboratory scene where we began our journey tonight. You stand once more in Cavendish Laboratory, watching final puzzle pieces assembling into beautifully coherent picture under Watson and Crick's intensely focused attention. Genetic code's elegant physical form crystallizing from X-ray crystallography. Diffraction patterns through patient night hours of quiet mathematical concentration and molecular model building. Architecture of heredity revealing itself at last through combination of Franklin's experimental data, Chargaff's base pairing rules, Pauling's alpha helix inspiration, and Watson-Crick's stereochemical reasoning double helix structure, about to transform biology forever by explaining simultaneously how genetic information stored and replicated. You stand once more in Cambridge Laboratory, spring evening 1953, watching James Watson and Francis Crick working late hours assembling their famous double helix model. Laboratory quiet, except for occasional brass molecular models clicking together as Watson tests different base pairing arrangements. Crick's slide rule calculations producing soft sliding sounds, distant rain continuing its steady rhythm against windows. Tobacco smoke drifts lazily from Crick's ever-present cigarette resting in ashtray. Desk lamp pools yellow light across photo 51, Rosalind Franklin's X-ray crystallography image showing unmistakable X-shaped diffraction pattern characteristic of helical structure. Watson measures distances between spots using ruler, calculating helical parameters. Pattern indicates two strands wound around each other in right-handed helix. Complete turn every 34 angstroms, containing 10 base pairs, each base pair separated by 3.4 angstroms vertically along helix axis. Diameter approximately 20 angstroms, consistent with sugar phosphate backbones on helix exterior, and nitrogenous bases pointing inward toward helix center. Critical breakthrough came from recognizing Chargaff's base pairing rules. Amount of adenine always equals thymine. Amount of guanine always equals cytosine across different species. Watson suddenly realizes bases must pair specifically. Adenine with thymine, guanine with cytosine, forming hydrogen bonds holding two complementary strands together. Adenine-thymine pairs form 2 hydrogen bonds. Guanine-cytosine pairs form 3 hydrogen bonds, both pairs coincidentally spanning identical width, approximately 11 angstroms, allowing regular helical geometry. This complementary base pairing immediately suggests copying mechanism. Each strand serves as template directing synthesis of complementary new strand, allowing precise hereditary information transmission across generations. Elegant simplicity of structure explaining simultaneously information storage through base sequence and information replication through template-directed synthesis. You watch Watson building physical model using brass plates representing bases, metal rods representing sugar phosphate backbones, carefully adjusting bond angles, checking stereochemical feasibility. Bases must fit within helix interior without steric clashes between atoms occupying same space. Hydrogen bonds between paired bases must align properly geometrically. Sugar phosphate backbones must follow smooth helical path without kinks or distortions. Model building requires spatial reasoning and chemical intuition, testing whether proposed structure physically possible given known atomic bond lengths, bond angles, van der Waals radii, preventing atoms approaching too closely. Each adjustment changes multiple parameters simultaneously. Rotating one base affects backbone trajectory, requiring compensating adjustments elsewhere, maintaining helical regularity. After hours of tinkering, model finally works perfectly. 2 anti-parallel strands running opposite directions. One strand oriented 5' to 3' going upward. Complementary strand oriented 3' to 5' going upward. Wind around common axis, forming right-handed double helix, with major and minor grooves visible between helical turns, where backbone approaches closer or farther from helix center. Base pairs stack parallel to each other, perpendicular to helix axis, stacking interactions between adjacent base pairs, contributing additional stability beyond hydrogen bonding between complementary bases. Watson and Crick immediately recognize revolutionary implications. Structure suggests copying mechanism so beautiful must be correct. Complementary base pairing means each strand contains information specifying complementary strand sequence, allowing accurate replication simply by separating strands and synthesizing new complementary strands using each original strand as template. Next morning, February 28, 1953, Watson and Crick walk to Eagle Pub for lunch, Crick famously announcing they have discovered secret of life. Perhaps overstatement, but double helix structure indeed transforms biology by revealing molecular basis of heredity. They publish findings April 25, 1953 in Nature Journal, remarkably brief 900-word paper titled Molecular Structure of Nucleic Acids, a structure for deoxyribose nucleic acid, accompanied by Rosalind Franklin and Morris Wilkins X-ray crystallography data supporting proposed structure. Understated final sentence of paper notes modestly. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Scientific understatement masking enormous significance. But proposing structure differs from proving structure correct, and demonstrating replication mechanism actually works as hypothesized. You drift forward 5 years, finding yourself at California Institute of Technology, Pasadena, year 1958. You stand in biochemistry laboratory of Matthew Meselson and Franklin-Stahl, young postdoctoral researchers designing elegant experiment testing DNA replication mechanism. Watson-Crick model predicts semi-conservative replication, where each original DNA strand serves as template for new complementary strand synthesis, resulting in two daughter DNA molecules, each containing one original parental strand plus one newly synthesized strand. Alternative models suggest conservative replication, where original double helix remains intact, an entirely new double helix synthesized, or dispersive replication, where DNA breaks into fragments and new DNA interspersed with old DNA throughout molecules. Meselson and Stahl's experimental strategy exploits nitrogen isotopes differing slightly in atomic mass. Normal nitrogen-14 predominates naturally, but heavy nitrogen-15 isotope available commercially substitutes for normal nitrogen in biological molecules, including DNA's nitrogenous bases. Bacteria grown for many generations in medium containing nitrogen-15 incorporate heavy nitrogen into all DNA synthesized, making DNA molecules denser than normal DNA containing nitrogen-14. Density difference seems trivial, perhaps 1%, but centrifugation in cesium chloride density gradient can separate DNA molecules differing slightly in density, allowing distinguishing between heavy DNA, light DNA, or hybrid DNA containing one heavy strand plus one light strand. You watch experimental procedure unfold across several days of careful work. First, Meselson cultures Escherichia coli bacteria in minimal medium, where nitrogen-15 labelled ammonium chloride provides sole nitrogen source. Bacteria grow for approximately 14 generations, sufficient time ensuring all cellular DNA fully labelled with heavy nitrogen-15, confirmed by extracting DNA and checking density through cesium chloride density gradient centrifugation. Then, at time 0, bacteria transferred abruptly to fresh medium containing normal nitrogen-14, allowing resumed growth using light nitrogen for all new DNA synthesis. Samples collected at intervals corresponding to bacterial generations, DNA extracted gently avoiding shearing long chromosomal DNA molecules, analysed by cesium chloride density, gradient ultracentrifugation. Centrifugation technique requires specialised equipment and meticulous technique. Cesium chloride solution prepared at concentration approximately 7.7 molar, nearly saturated, very dense clear liquid. Small amount of purified DNA added to cesium chloride solution in special ultracentrifuge tube made of transparent plastic allowing light passage. Tube sealed carefully, loaded into angle head rotor of Spinco Model E analytical ultracentrifuge. Sophisticated instrument allowing observation of samples during centrifugation through optical system. Rotor spun at tremendous speed, 44,770 revolutions per minute, generating centrifugal force approximately 140,000 times Earth's gravity. Enormous force causing cesium chloride forming smooth density, gradient from tube top to bottom, heavy cesium chloride sedimenting toward tube bottom, while water concentrates toward top, creating continuous gradient. DNA molecules migrate within gradient until reaching position where cesium chloride density equals DNA density, forming narrow band visible through ultraviolet light absorption. Centrifugation continues for approximately 20 hours, allowing equilibrium establishment. Cesium gradient forms fully. DNA migrates to equilibrium density position. Then Meselson and Stahl photograph tubes using ultraviolet light, DNA absorbing strongly at 260 nanometers, appearing as dark bands against lighter background on photographic film. Positions of DNA bands relative to tube length indicates DNA density directly. Heavy nitrogen 15, DNA bands near tube bottom where cesium chloride density matches. Heavy DNA density approximately 1.724 grams per cubic centimeter. Light nitrogen 14. DNA bands higher in tube where density approximately 1.710 grams per cubic centimeter. Hybrid DNA containing one heavy strand, one light strand bands at intermediate position approximately 1.717 grams per cubic centimeter, halfway between fully heavy and fully light. Results beautifully clear and unambiguous. Initially, before transfer to light medium, all DNA shows single band at heavy position as expected from complete nitrogen 15 labeling. After one generation growth in light medium, DNA shows single band at hybrid position, demonstrating newly replicated DNA contains one heavy original strand plus one light newly synthesized strand, exactly as semi-conservative replication predicts. After two generations, DNA shows two bands, one at hybrid position, one at light position, in approximately equal proportions, again precisely matching semi-conservative model predictions, where hybrid molecules from first generation serve as templates producing one hybrid molecule plus one fully light molecule. Conservative replication would produce only heavy and light DNA, never hybrid. Dispersive replication would produce gradual density shift rather than discrete bands. Only semi-conservative replication produces observed banding pattern. Experiment definitively proves Watson-Crick replication hypothesis correct. You drift forward 3 more years, finding yourself at National Institutes of Health, Bethesda, Maryland, year 1961. You stand in biochemistry laboratory of Marshall Nirenberg and Heinrich Mattei, researchers attempting to decipher genetic code, relationship between DNA nucleotide sequences and amino acid sequences in proteins. Francis Crick proposed 1957 that genetic information flows from DNA to RNA to protein, central dogma of molecular biology, but exact coding relationship remains mysterious. 20 amino acids commonly found in proteins must be specified by 4 DNA bases, suggesting combinations of bases forming codons encoding amino acids. Simple mathematics shows single bases cannot specify 20 amino acids, only 4 possible, and 2 base codons insufficient, only 16 possible combinations. Therefore, minimum 3 bases per codon required, giving 64 possible triplet combinations, more than sufficient encoding 20 amino acids, with redundancy allowing multiple codons specifying same amino acid. Nirenberg and Mattei develop ingenious cell-free protein synthesis system allowing testing codon assignments experimentally. System consists of bacterial cell extract containing ribosomes, transfer RNAs, amino acids, necessary enzymes, and cofactors for translation, but lacking DNA and messenger RNA. Adding synthetic RNA polymer to system programs, ribosomes synthesizing specific polypeptide depending on RNA sequence. Simplest experiment uses synthetic polyuridylic acid, poly-U, RNA polymer, containing only uracil bases in long repeating chain, synthesized in vitro using purified enzyme polynucleotide phosphorylase. Poly-U added to cell-free extract along with mixture of 20 amino acids, 1 amino acid radioactively labeled, allowing detecting which amino acid incorporated into synthesized polypeptide. You watch Nirenberg pipetting carefully at laboratory bench, working under fume hood for safety when handling radioactive materials. He prepares cell-free extract by growing bacteria overnight in rich medium, harvesting by centrifugation, breaking cells open using French press, forcing bacterial suspension through small orifice at high-pressure shearing membranes, clarifying lysate by centrifugation, removing cell debris and unbroken cells, saving supernatant containing soluble components. Extract supplemented with ATP, providing energy, GTP serving as energy source and ribosome factor, magnesium ions required for ribosome stability and activity, potassium ions maintaining proper ionic strength. Amino acid mixture prepared with 19 amino acids unlabeled plus 1 amino acid labeled with radioactive carbon-14, allowing detection of incorporated radioactivity if that specific amino acid incorporated into protein. Small aliquots of cell-free extract dispensed into test tubes on ice, keeping reaction components inactive until warmed to physiological temperature. Synthetic poly-URNA added at known concentration, tubes transferred to 37-degree water bath, initiating protein synthesis. After 30-minute incubation, allowing sufficient time for translation, reactions stopped by adding trichloroacetic acid, precipitating proteins, but leaving unincorporated free amino acids soluble. Precipitated proteins collected on filter paper discs, washed extensively with trichloroacetic acid, removing unincorporated amino acids, dried radioactivity measured using liquid scintillation, counter-detecting beta particles emitted by carbon-14 decay. Results dramatic and clear-cut. When poly-URNA added to extract containing radioactive phenylalanine, enormous radioactivity incorporation occurs, synthesizing polyphenylalanine, containing hundreds of phenylalanine residues linked together. When any other radioactive amino acid used instead, virtually no radioactivity incorporation detected, demonstrating poly-U specifically encodes phenylalanine, not other amino acids. Therefore, triplet codon UUU specifies phenylalanine. First codon assignment deciphered. Following similar strategy, Nirenberg, Mattei, and collaborating scientists synthesize other simple RNA polymers, testing codon assignments systematically. Poly-A encodes polylysine, revealing AAA codes, lysine. Poly-C encodes polyproline. CC codes proline. Random copolymers containing multiple bases in random sequences produce multiple amino acids incorporated in proportions predictable from codon frequencies, allowing deducing additional assignments. Severo Ochoa's laboratory at New York University and Har Gobind, Khorana's laboratory at University of Wisconsin synthesized defined sequence RNA oligonucleotides, testing specific codon assignments directly. By 1966, entire genetic code table complete. 61 codons specify amino acids, 3 codons serve as stop signals terminating translation. Genetic code reveals elegant properties. Code redundant. Most amino acids specified by multiple synonymous codons, differing usually in third position, wobble position, where base pairing rules relaxed, allowing non-Watson-Crick pairing. Code nearly universal across all organisms from bacteria to humans, suggesting code established early in evolution before divergence of major lineages, allowing genetic engineering moving genes between species. Code organized such that chemically similar amino acids, often specified by related codons, minimizing effects of point mutations, typically changing single nucleotide producing codon, differing by one position, often resulting in amino acid substitution with similar chemical properties preserving protein function. DNA replication seems flawless, copying billions of base pairs each cell division, yet every replication introduces tiny random mutations, providing raw material for evolution. nature's way keeping things interesting without rewriting entire genetic program every generation. Error rate remarkably low, approximately 1 error per billion. Bases copied, achieved through multiple proofreading mechanisms including DNA polymerase, 3' to 5' exonuclease activity, removing misincorporated nucleotides immediately. Mismatch repair systems scanning newly replicated DNA, correcting errors missed by polymerase. Yet occasional errors persist, accumulating gradually over generations, driving evolutionary change, sometimes causing genetic diseases when occurring in critical gene sequences. But reading genetic code one codon at a time proves extremely laborious. Determining complete gene sequences, let alone entire genome sequences, requires revolutionary technical advances in DNA sequencing methodology. You drift forward to 1977, finding yourself at Medical Research Council Laboratory of Molecular Biology, Cambridge, England, where Frederick Sanger, British biochemist previously awarded Nobel Prize for determining insulin amino acid sequence, develops new DNA sequencing method enabling reading hundreds of bases continuously. Sanger's chain termination method exploits modified nucleotides called dideoxy nucleotides lacking 3' hydroxyl group normally present on deoxyribonucleotides. During DNA synthesis, DNA polymerase adds nucleotides sequentially to growing chain, forming phosphodiester bond between 5' phosphate of incoming nucleotide and 3' hydroxyl of previous nucleotide. If dideoxy nucleotide incorporated instead, lacking 3' hydroxyl group, prevents adding subsequent nucleotide, chain terminates at that position, including small fraction of dideoxy nucleotides in DNA synthesis. Reaction mixture along with normal deoxynucleotides produces mixture of terminated fragments of different lengths, each ending wherever dideoxynucleotide incorporated by chance. You watch careful sequencing procedure. Template DNA to be sequence prepared as single-stranded form, short synthetic oligonucleotide primer annealed to complementary region, providing 3' hydroxyl for initiating synthesis. 4 separate reactions prepared, each containing template primer complex, DNA polymerase enzyme, all 4 normal deoxynucleotides, DATP, DCTP, DGTP, DTTP, plus single dideoxynucleotide species in limiting amount. First reaction contains DDATP, causing chain termination specifically at positions where adenine incorporated. Second reaction contains DDCTP, terminating at cytosine positions. Third, DDGTP for guanine. Fourth, DDTTP for thymine. Each reaction produces nested set of terminated fragments sharing common starting point at primer but ending at different positions depending on where dideoxynucleotide randomly incorporated. Original Sanger method uses radioactive labeling for fragment detection, incorporating radioactive phosphorus-32 into synthesized DNA by including alpha-32P labeled DNTPs in reaction mix, labels newly synthesized fragments, allowing detection by autoradiography. After synthesis reactions complete, fragments separated by size using polyacrylamide gel electrophoresis. You watch careful gel preparation, dissolving acrylamide monomer with crosslinker bisacrylamide in buffer, adding polymerization catalyst TEMED and ammonium persulfate, initiating free radical polymerization forming gel matrix with precise pore sizes separating DNA fragments differing by single nucleotide. Gel poured between glass plates separated by thin spacers, creating gel slab approximately 1 mm thick, comb inserted at top creating sample wells. After polymerization complete, gel loaded into vertical electrophoresis apparatus filled with buffer, samples loaded into wells alongside size markers. Electric current applied across gel, voltage typically 50 volts per centimeter, gel length generating electric field. DNA fragments, negatively charged due to phosphate groups, migrate toward positive electrode, smaller fragments moving faster through gel pores than larger fragments, separating by size with single nucleotide resolution. Electrophoresis continues several hours, allowing fragments separating across gel length. Then gel carefully removed from glass plates, placed on filter paper, covered with plastic wrap, dried under vacuum at moderate temperature. Dried gel placed against X-ray film in darkroom, stored at -80 degrees Celsius for 1 to several days, allowing radioactive decay, exposing film. Film developed, revealing ladder-like pattern of dark bands representing terminated fragments. Reading sequence requires comparing 4 lanes containing A, C, G, T termination reactions run side by side on same gel. Smallest fragments, fastest migrating, appear near gel bottom, largest, slowest fragments near top. Reading from bottom upward, identifying which lane contains band at each position, reveals sequence directly. If bottom band appears in A lane, first nucleotide is A. If next band up appears in T lane, second nucleotide is T. Continuing upward, systematically reading lane positions reconstructs complete DNA sequence 1 nucleotide at a time. Typical gel read approximately 500 bases before bands become too compressed at gel top for accurate interpretation. Later improvements replace radioactive detection with fluorescent dyes. Different coloured fluorescent dyes attached to dideoxy nucleotides, allowing running all 4 termination reactions in single tube and single gel lane. Automated DNA sequencers use laser excitation, causing fluorescent labelled fragments, emitting different colours detected by photomultiplier or CCD camera computer software, automatically calling bases from fluorescent signal traces, producing digital sequence readout. These automated systems revolutionize sequencing throughput, enabling ambitious genome sequencing projects determining complete DNA sequences of organisms from bacteria to humans. Yet sequencing requires substantial DNA quantities, typically micrograms of pure template. Obtaining sufficient DNA often proves limiting step, especially for precious samples like forensic evidence, ancient DNA from archaeological specimens, or clinical biopsies containing limited material. You drift forward 6 years, finding yourself on California Coastal Highway during spring evening 1983, inside car driven by Kari Mullis, biochemist working at Cetus Corporation Biotechnology Company. Mullis driving from Berkeley toward Mendocino weekend cabin, mentally working through molecular biology problem while girlfriend sleeps in passenger seat. Suddenly inspiration strikes. Elegant method for exponentially amplifying specific DNA sequences through repeated cycles of heat denaturation and enzymatic synthesis. Polymerase chain reaction, PCR, concept deceptively simple but revolutionary in impact. Idea based on DNA's complementary structure and temperature-dependent behavior. DNA double helix, stable at moderate temperatures, but denatures into single strands when heated above approximately 94 degrees Celsius. Hydrogen bonds between base pairs breaking reversibly. Upon cooling, complementary single strands reanneal, reforming double helix through Watson-Crick base pairing. If short synthetic oligonucleotide primers present complementary to regions flanking target DNA sequence, primers anneal specifically to template strands at lower temperature around 55 degrees Celsius. DNA polymerase enzyme then extends primers, synthesizing new complementary strands at moderate temperature around 72 degrees Celsius. Repeating this thermal cycle, denature at high temperature, anneal primers at intermediate temperature extend with polymerase at moderate temperature. Doubles target DNA amount each cycle. 30 cycles produces theoretical billion-fold amplification. You find yourself transported to Cetus Corporation Molecular Biology Laboratory, watching PCR development from concept to practical reality. Initial experiments use E. coli DNA polymerase, requiring adding fresh enzyme each cycle, because heat denaturation denatures not only template DNA, but also polymerase enzyme. Process tedious, requiring opening reaction tubes, adding enzyme after every cycle, risking contamination, limiting automation potential. Breakthrough comes from using thermostable DNA polymerase, isolated from Thermus aquaticus, thermophilic bacterium living in hot springs at Yellowstone National Park, discovered by Thomas Brock in 1960s. Taq polymerase, as enzyme known, remains active after exposure to 94 degrees Celsius, allowing enzyme persisting through many thermal cycles without degradation or inactivation. You watch technicians setting up PCR reaction at laboratory bench. Small, thin-walled plastic tube holds 25 microliter reaction volume, about size of small raindrop. Reaction mixture contains template DNA to be amplified, present in vanishingly small amounts, perhaps just few hundred copies, or even single molecule. Pair of synthetic oligonucleotide primers, typically 20 nucleotides long designed to flank target region, complementary to opposite DNA strands. 4 deoxyribonucleotide triphosphates, DATPD, CTPD, GTPD, TTP, providing building blocks for synthesis, magnesium chloride required as DNA polymerase cofactor, buffered solution maintaining proper pH and ionic strength, and thermostable Taq polymerase enzyme. Tube capped securely, preventing evaporation during heating, placed in thermal cycler instrument, programmed with specific temperature time profile. Thermal cycler consists of metal heating block containing array of wells precisely fitting PCR tubes, powerful Peltier thermoelectric heating cooling elements allowing rapid temperature changes, precise electronic temperature control, maintaining set point within tenths of degree, heated lid pressing onto tube caps preventing condensation. You watch instrument running through programmed cycle profile displayed on digital screen. Initial denaturation step 94 degrees Celsius for 2 minutes ensures complete separation of all template double helices into single strands. Then cycling begins. Denaturation, step 94 degrees Celsius for 30 seconds, melts all double-stranded DNA, annealing step typically 55 degrees Celsius, though optimal temperature depends on primer sequences. For 30 seconds allows primers binding specifically to complementary template regions. Extension step 72 degrees Celsius for 1 minute per kilobase target length allows Taq polymerase synthesizing new strands at optimal activity temperature. Cycle repeats typically 30 times, taking approximately 2 to 3 hours total, depending on target size and instrument ramping speed. Laboratory atmosphere during PCR run quiet and patient. Thermal cycler hums softly, electronic temperature display cycling between programmed values, faint clicking sounds as Peltier elements switch between heating and cooling modes. Technicians continue other work while PCR runs unattended. Instrument automation allowing multiple samples processing simultaneously without constant monitoring. After cycling completes, final extension step 72 degrees Celsius for 5 minutes ensures all partial synthesis products completely extended, then reactions cool to 4 degrees Celsius for storage. Success assessed by gel electrophoresis, analysing PCR products. A small aliquot of completed reaction loaded onto agarose gel, lower percentage gel, suitable for larger DNA fragments, unlike high percentage polyacrylamide gels used for sequencing. Gel prepared by melting agarose powder in buffer using microwave oven, pouring molten agarose into casting tray with comb creating sample wells, allowing solidification at room temperature, forming firm translucent gel. Gel placed in electrophoresis chamber filled with buffer, samples mixed with loading dye containing glycerol for density plus tracking dyes, bromophenol blue and xylene cyanol showing migration progress loaded into wells using pipette. Electric current applied, DNA migrates toward positive electrode, separated by size with larger fragments migrating slower than smaller fragments. After electrophoresis, approximately 45 minutes, gel removed from chamber, stained with ethidium bromide fluorescent dye, intercalating between DNA base pairs, visualized under ultraviolet transilluminator. Successful PCR produces bright fluorescent band at expected size corresponding to distance between primers. Band intensity indicating amplification, yield typically producing micrograms of product from femtograms of starting template, million-fold amplification, allowing detecting single DNA molecule in complex sample. Failed reactions show no band or non-specific bands at wrong sizes from primer, misdirected amplification, or primer-dimer artifacts, where primers amplify each other rather than template. PCR revolutionizes molecular biology, making genetic analysis accessible far beyond specialized laboratories. Applications multiply rapidly. Forensic DNA profiling from minute blood or tissue samples, paternity testing, ancient DNA analysis from museum specimens or fossil remains, detection of infectious disease pathogens, prenatal genetic diagnosis, cancer mutation screening, evolutionary studies comparing homologous genes across species. Before PCR, such analyses required substantial, pure DNA quantities obtainable only through extensive molecular cloning, growing recombinant bacteria, purifying plasmid DNA, laborious processes taking weeks. After PCR, same analyses achievable in hours from crude samples without purification or cloning. You drift forward several years watching PCR technology diversify into variants serving specialized applications. Reverse transcription. PCR measures RNA expression levels by first copying RNA into complementary DNA using reverse transcriptase enzyme before PCR amplification. Real-time quantitative PCR monitors. Amplification product accumulation during cycling using fluorescent dyes or probes, allowing precise quantification of starting template amounts through cycle threshold values, where fluorescence exceeds background, revolutionizing gene expression studies by replacing laborious northern blots and RNase protection assays. Long-range PCR using specialized high-fidelity polymerase mixtures amplifies targets exceeding 20 kilobases previously inaccessible to standard PCR. Multiplex PCR amplifies multiple targets simultaneously using multiple primer pairs in single reaction, enabling complex genetic analyses from limited samples. Laboratory routines adapt to ubiquitous PCR usage. Dedicated rooms separate pre-PCR from post-PCR areas, preventing amplicon contamination, enormous amplification power meaning single molecule contaminating. New reaction produces false positive results. Careful technique prevents contamination. Filtered pipette tips prevent aerosol cross-contamination. Frequent glove changes reduce transfer. Separate reagent aliquots for each experiment. Prevent batch contamination, UV irradiation of work surfaces, and reagents damages contaminating DNA, rendering it non-amplifiable. Regular cleaning with bleach, destroying contaminating nucleic acids. Regular cleaning. Thermal cyclers become laboratory workhorses, multiple instruments running continuously, familiar background hum, and temperature cycling part of daily laboratory soundscape. PCR setup procedures become routine muscle memory. Retrieving frozen reagents from -20 freezer, thawing on ice, vortexing to mix, brief centrifugations spinning down droplets, pipetting master mix, containing all common components, aliquoting into individual tubes, adding templates last, preventing contamination, capping, loading into thermal cycler, starting programmed profile, washing work surface, disposing of waste appropriately, returning reagents to freezer. Process so familiar, technicians work efficiently while thinking about experimental design, anticipated results, next steps. You watch graduate student late evening working alone in quiet laboratory. Thermal cycler running in background, faint hum, an LED display showing remaining cycles counting down. Student sits at desk nearby, analysing previous gel images on computer screen, comparing band intensities, calculating ratios, entering data into spreadsheet. Occasional glance toward thermal cycler checking progress. Laboratory, comfortable temperature. Overhead fluorescent lights reflecting off black laboratory benches. Fume hood, motor providing white noise. Solitary focused work atmosphere common in research laboratories after regular hours when interruptions minimal and concentration deep. Student's notebook open beside computer showing careful records. Dates, reagent lot numbers, reaction compositions, cycling parameters, gel photographs, results interpretations, next experiments planned. Thermal cycler beeps softly, indicating cycling complete. Student walks to instrument, opens heated lid, releasing warm air, carefully removes tubes from block still warm to touch, places in ice bucket. Prepares loading gel while tubes cool, retrieves previously cast gel from cold room where stored in sealed container with buffer preventing drying, places in electrophoresis chamber, adds buffer to chamber submerging gel, loads molecular weight ladder in first lane, mixes samples with loading dye on parafilm sheet, using separate pipette tip for each sample. Preventing cross-contamination, loads samples sequentially in adjacent wells, connects electrodes, sets power supply to constant voltage, typically 100 volts, starts electrophoresis timer. Sits back watching blue tracking dye begin migrating slowly through gel, takes notes in laboratory notebook about samples loaded and expected results. After 45 minutes, stops electrophoresis, carefully removes gel from chamber, carrying in staining tray to fume hood where ethidium bromide staining performed, adds ethidium bromide solution diluted in water, incubates 15 minutes with gentle shaking, pours off ethidium bromide into designated waste container, rinses gel extensively with water, removing unbound dye, reducing background fluorescence, transfers gel to UV transilluminator, closes protective hood blocking ultraviolet exposure, switches on UV light. Gel glows orange through amber filter. DNA bands appear as bright orange fluorescent bars against darker background. Student examines pattern noting, successful amplification products at expected sizes, no primer-dimer artifacts near bottom. Clean specific amplification confirming primers worked properly. Presses button. Capturing digital image, saves image file with descriptive name, including date and experiment number, prints copy for laboratory notebook, switches off UV light. Results match predictions. Target gene amplified successfully from all treated samples, absent from negative controls confirming specificity, band intensities varying between conditions, suggesting differential expression requiring quantification. Student makes notes, interpreting results, considers whether repeating experiment with more replicates or proceeding to next planned experiment, feels satisfaction from successful result validating previous days troubleshooting, adjusting primer concentrations, and annealing temperature optimizing specificity. Late hour now, nearly midnight, time stopping for evening. Powers down equipment, disposes of waste appropriately. Ethidium contaminated materials in designated hazardous waste container. General plasticware in biohazard bins. Washes hands thoroughly. Turns off most lights, leaving one on for safety. Locks laboratory door. Heads home through quiet campus streets. Such scenes repeat nightly in laboratories worldwide. PCR's democratization of genetic analysis, enabling researchers addressing questions previously inaccessible. Before PCR, genetic analysis required extensive training, specialized equipment, substantial funding, restricted genetic research to well-equipped laboratories. After PCR, basic genetic analysis possible with modest thermal cycler costing few $1,000s, standard laboratory equipment, commercially available reagents, enabling researchers everywhere contributing to genetic knowledge, expansion accelerating biological understanding dramatically through distributed accessible technology. Mullis awarded Nobel Prize in Chemistry 1993. Recognizing PCR's transformative impact on molecular biology and beyond into medicine, forensics, agriculture, evolutionary biology, all fields touching genetic information. Forensic applications particularly transformative. Traditional forensic blood typing requires substantial fresh samples, provides limited discrimination between individuals, easily degraded by environmental exposure. PCR-based DNA profiling analyzes specific highly variable chromosomal regions called short tandem repeats, STRs, where individuals differ in number of repeated sequence motifs, requires only nanograms of DNA recoverable from dried bloodstains, saliva on cigarette butts, hair roots, touched objects leaving skin cells. Combined analysis of multiple STR loci provides identification probabilities exceeding 1 in billion, effectively unique individual identification from minute samples. Cold cases solved decades after crimes through DNA analysis of archived evidence samples. Innocence Project uses DNA testing, exonerating wrongly convicted individuals, over 375 exonerations in United States alone through 2020, demonstrating both power of DNA evidence and fallibility of other identification methods like eyewitness testimony. Ancient DNA research enabled by PCR's extreme sensitivity amplifying degraded DNA fragments. Archaeological specimens thousands of years old preserve DNA in damaged fragmented form, long molecules broken into short pieces through chemical degradation, strand breaks accumulating, bases modified through oxidation and hydrolysis. Standard molecular cloning requires intact long molecules impossible from ancient samples. PCR amplifies short fragments successfully despite damage, allowing genetic analysis of extinct species, ancient human populations, evolutionary relationships. Neanderthal genome sequenced from 40,000-year-old bone specimens, revealing interbreeding with modern humans, Neanderthal DNA persisting in non-African human populations at approximately 2%. Denisovan hominin identified solely from DNA extracted from single finger bone found in Siberian cave. Population movements and migrations reconstructed from ancient DNA showing prehistoric demographic changes invisible to archaeology alone. Clinical diagnostics revolutionized by PCR detecting pathogens directly from patient samples. Traditional bacterial culture requires days, growing bacteria on selective media, fungal cultures weeks, viral culture difficult or impossible for many viruses. PCR detects pathogen-specific DNA sequences within hours, providing rapid diagnosis, guiding appropriate treatment. HIV infection detected weeks earlier using PCR than antibody tests, critical window for preventing transmission and starting antiretroviral therapy. Drug-resistant mutations identified directly from clinical samples, guiding antibiotic selection. Cancer mutations detected from tumour biopsies or liquid biopsies analysing circulating tumour DNA in blood, enabling precision medicine matching treatments to tumour molecular profiles. Prenatal diagnosis screens fetal DNA circulating in maternal blood for chromosomal abnormalities, avoiding invasive amniocentesis procedures risking miscarriage. You drift forward through time, finding yourself amid ambitious international collaboration. Year 1990, Human Genome Project officially launching with goal determining complete DNA sequence of human genome. Approximately 3 billion base pairs distributed across 23 chromosome pairs. Project represents biological equivalent of Apollo Moon program in scope, ambition, technological challenge, requiring innovations in sequencing technology, computational methods, international coordination. Initial estimates suggest 15-year timeline, $3 billion budget, $1 per base pair, staggering investment justified by transformative medical and scientific potential. Skeptics question feasibility, comparing task to attempting reading encyclopedia letter by letter using magnifying glass. But advocates argue technological improvements during project execution will enable completion within budget and timeline. You stand in Genome Sequencing Center, large open laboratory space filled with dozens of automated DNA sequencers, machines running continuously day and night producing sequence data. Capillary electrophoresis sequencers replace earlier slab gel systems using thin glass capillaries instead of polyacrylamide gel slabs, allowing higher throughput with automated sample loading, electrophoresis, detection without manual gel handling. Each sequencer contains 96 capillaries running parallel, analysing 96 samples simultaneously, completing runs in 2 hours, producing approximately 500 bases per capillary, yielding roughly 48,000 bases per instrument per run. Laboratory houses 30 instruments producing over 1 million bases daily, substantial output, yet representing tiny fraction of 3 billion base total, requiring thousands of instrument days accumulating over years. Laboratory atmosphere distinctly industrial compared to individual investigator laboratories pursuing focused research questions. Technicians work at stations preparing samples for sequencing, pipetting bacterial cultures growing overnight in 37-degree shaking incubators into 96 well microplates, extracting plasmid DNA using silica membrane mini-prep columns binding DNA selectively while washing away proteins and RNA contaminants, eluting purified DNA in sterile water or buffer. Conducting sequencing reactions, mixing template, plasmid, DNA with sequencing primers, complementary to vector sequences, flanking inserted genomic fragments, adding DNA polymerase enzyme. 4 fluorescent dideoxy nucleotides, each labelled with different coloured dye, allowing 4-colour detection. Buffer providing optimal pH and ionic strength. Dispensing reactions into thin-walled PCR plates suitable for thermal cycling. Loading plates into thermal cyclers for sequencing reaction temperature cycling. Initial denaturation at 96 degrees Celsius, melting double-stranded template, followed by 25 cycles of denaturation at 96 degrees, annealing at 50 degrees allowing primers binding, extension at 60 degrees permitting polymerase synthesizing complementary strand until randomly incorporating fluorescent dideoxy nucleotide terminating chain. After cycling complete, removing unincorporated dye-labeled terminators through ethanol precipitation or spin column purification, preventing high fluorescent background obscuring sequence signal, resuspending purified products in formamide loading buffer, transferring into special sequencing plates compatible with automated capillary sequencers. Automated liquid handling robots perform repetitive pipetting tasks, robotic arms moving between positions, multi-channel pipetting heads, simultaneously aspirating and dispensing from multiple wells, reducing human error from fatigue during hundreds of repetitive transfers, increasing throughput consistency and reproducibility, preventing repetitive strain injuries affecting technicians performing manual pipetting 8 hours daily. Robots programmed through computer interface specifying source plates, destination plates, volumes, speeds, ensuring precise reproducible liquid transfers. Refrigerators line walls storing temperature-sensitive reagents. -20 degree freezers hold enzymes and fluorescent nucleotides. -80 degree freezers preserve bacterial glycerol stocks, maintaining clones for future use. Large incubators hold hundreds of bacterial culture flasks, growing transformed bacteria containing genomic clones. Centrifuges spin plates separating cells from medium during DNA extraction. Boxes of pipette tips, microplates, reagent bottles stack on shelves above benches. Sequencing instruments themselves occupy considerable laboratory space, each sequencer roughly refrigerator-sized, containing automated capillary array, laser excitation system, 4-color fluorescence detection optics, sophisticated electronics, computer control systems. Capillaries, thin fragile glass tubes, approximately 50 centimeters long, 50 micrometers internal diameter, filled with polymer matrix separating DNA fragments by size as electric current applied. Each instrument contains an array of 96 capillaries running parallel, automated systems injecting samples electrokinetically into capillaries, applying high voltage across capillaries, driving negatively charged DNA fragments migrating toward positive electrode, detecting fluorescent signals as fragments pass detection window near capillary end where focused laser beam excites fluorophores. Emitted light collected through microscope objective, spectrally separated by dichroic mirrors and filters into 4 detection channels corresponding to 4 dye colors, photomultiplier tubes converting light to electrical signals, analog to digital converters sampling signals thousands of times per second, generating digital traces showing fluorescence intensity over time. Data collection software displays real-time electropherograms as sequencing runs progress, 4 color traces showing peaks representing bases passing detector, green peaks for adenine, blue for cytosine, black for guanine, red for thymine, peaks separated temporally based on fragment size, automated base calling algorithms analyzing peak spacing, height, width, assigning most probable base at each position, quality scores indicating confidence in each base, call based on signal strength and peak resolution. Each capillary generates approximately 500 base sequence, extending from sequencing primer into insert, forward, and reverse reads from opposite fragment ends, providing overlapping sequences enabling assembly. Complete sequencing run takes approximately 2 hours, after which capillaries automatically cleaned by flushing polymer, instruments reload samples from next plate, continuous operation allowing 6 to 8 runs per 24 hours, generating approximately 50,000 bases per instrument daily. Computers continuously collect data from sequencers, storing raw fluorescence traces and processed sequence files, transferring files to central servers for storage and downstream analysis. Network infrastructure handles large data volumes, terabytes of sequence data, accumulating monthly, requiring robust storage systems and backup procedures preventing catastrophic data loss. Database systems track sample identities, clone information, sequencing reads, assembly status, maintaining complex relationships between physical samples and digital data. Sequencing strategy employs shotgun approach, fragmenting chromosomes randomly into smaller pieces, cloning fragments into bacterial vectors, sequencing fragment ends from both directions, generating paired reads, computationally assembling overlapping fragments into complete continuous sequences called contigs. Random fragmentation means same genomic regions sequenced multiple times from different fragments, providing redundancy, confirming accuracy, coverage depth, typically 5 to 10-fold, ensuring each base sequenced multiple times, reducing errors. Assembly computationally intensive, requiring sophisticated algorithms identifying overlapping sequences, resolving repetitive regions, appearing multiple genomic locations, potentially causing misassembly, ordering and orienting contigs into chromosome-length scaffolds. You watch bioinformatician at computer workstation analyzing sequence assemblies, screen displaying contig alignment visualization, showing how sequenced fragments overlap, gaps requiring additional sequencing, potential misassemblies where sequences don't align properly, suggesting errors needing resolution. Genome assembly resembles jigsaw puzzle with 3 billion pieces, many pieces nearly identical, making matching difficult. Some regions missing from sequencing, requiring targeted finishing work. Assembly software runs on powerful computer clusters, multiple processors working parallel, analyzing millions of sequences, identifying overlaps, building consensus sequences, iteratively refining assemblies as new data accumulated. Project employs hierarchical shotgun strategy, first mapping chromosomes into large overlapping clones contained in bacterial artificial chromosomes. BACs holding approximately 150 kilobase inserts, creating physical map showing BAC locations along chromosomes through fingerprinting and end sequencing, then shotgun sequencing individual BACs separately and finally assembling BAC sequences into complete chromosomes. This clone-by-clone approach provides intermediate scaffolds simplifying final assembly compared to whole genome shotgun approach, sequencing entire genome at once attempted by competing private company Celera Genomics, founded by Craig Venter. Draft sequence released June 2000 in joint announcement by President Bill Clinton and Prime Minister Tony Blair, public consortium, and Celera simultaneously publishing draft sequences in Nature and Science journals, February 2001. Coverage approximately 90% of genome, with some gaps remaining in highly repetitive regions like centromeres and telomeres, proving difficult to sequence and assemble. Finished complete sequence published April 2003, 50 years after Watson-Crick double helix. Structure determination 99% complete, accuracy exceeding 99.99%, one error per 10,000 bases, representing culmination of thousands of scientists' efforts across dozens of institutions worldwide. Sequence reveals surprising findings. Genome contains approximately 20,000 protein-coding genes, far fewer than 50 to 100,000 previously estimated, suggesting human complexity arises not from gene number, but from sophisticated gene regulation, alternative splicing, generating multiple protein variants from single genes, complex interaction networks between gene products. Large fraction of genome consists of repetitive elements, transposable elements comprising 45%, sequences capable copying themselves to new genomic locations, evolutionary relics of ancient viral insertions, or selfish genetic elements propagating through genomes. Only approximately 1.5% of genome codes for proteins, remaining non-coding regions initially dismissed as junk DNA, but increasingly recognized as containing regulatory elements, long, non-coding RNAs, structural sequences organizing chromosomes, evolutionarily conserved sequences suggesting functional importance. Genome sequence enables systematic disease gene discovery. Comparing genomes from affected and unaffected individuals identifies genetic variants associated with diseases. Genome-wide association studies testing millions of genetic markers across thousands of individuals revealing susceptibility, loci for diabetes, heart disease, schizophrenia, countless conditions with genetic components. Rare disease diagnosis revolutionized through whole genome sequencing, identifying causative mutations in obscure genes, ending diagnostic odysseys for families seeking explanations for mysterious conditions. Pharmacogenomics personalizes medicine matching drug treatments to individual genetic variants affecting drug metabolism, efficacy, side effects, avoiding adverse reactions, and optimizing therapeutic responses. You drift forward, finding yourself in different laboratory. Year 2012, University of California, Berkeley, laboratory of Jennifer Doudna and Emmanuel Charpentier, biochemists investigating bacterial immune system called CRISPR, clustered regularly interspaced, short palindromic repeats. Bacteria use CRISPR-Cas systems defending against viral infections, storing viral DNA sequences in CRISPR arrays, serving as immunological memory, expressing these sequences as RNA guides, directing Cas nuclease enzymes, cutting matching viral DNA when virus reinfects, destroying invader before it replicates. Researchers recognize this molecular scissors system could be reprogrammed targeting any desired DNA sequence, creating universal programmable gene editing tool, revolutionizing biological research and therapeutic potential. You watch Laboratory Bench, where postdoctoral researcher Martin Jinek works carefully pipetting at Laboratory Bench, testing whether simplified CRISPR-Cas9 system works for gene editing in vitro. Natural bacterial system uses two separate RNA molecules, CRISPR RNA specifying target sequence and transactivating CRISPR RNA providing structural scaffold recruiting Cas9 nucleus. Jinek engineers single guide RNA combining both functions into single molecule, 20 nucleotide sequence complementary to target DNA sequence, fused to Cas9 binding scaffold, creating simpler two-component system. Guide RNA plus Cas9 protein. If design works, changing 20 nucleotide target sequence retargets system to any desired genomic location simply by synthesizing new guide RNA, no protein engineering required, dramatically simplifying gene editing compared to previous technologies like zinc finger nucleases or talons requiring custom protein design for each target. Laboratory equipped with standard molecular biology equipment for biochemical experiments testing protein-RNA interactions. Bacterial cultures grow in shaking incubator at 37 degrees Celsius, producing Cas9 protein expressed from cloned gene inserted into expression plasmid under control of inducible promoter, adding IPTG inducer compound triggers protein production. After overnight growth, bacteria harvested by centrifugation, cell pellets frozen at -20 degrees, facilitating cell lysis, thawed and resuspended in lysis buffer containing protease inhibitors preventing protein degradation, lysozyme enzyme digesting bacterial cell walls, detergents solubilizing membranes. Lysate clarified by high-speed centrifugation, separating soluble proteins from insoluble debris and inclusion bodies. Protein purified using chromatography columns, separating molecules based on chemical properties. Bacterial lysate loaded onto nickel, affinity column, glass column packed with nickel chelating resin, binding proteins containing histidine tags, short sequences of 6 consecutive histidine residues engineered onto Cas9's N-terminus, or C-terminus, specifically for purification purposes. Histidine residues coordinate nickel ions through imidazole side chains, capturing tagged protein, while untagged bacterial proteins wash through column unbound. After extensive washing with buffer containing low imidazole concentration, displacing weakly bound contaminants while maintaining Cas9 binding, elution buffer with high imidazole concentration competes with histidine tag for nickel binding, releasing purified Cas9 flowing through column collected in fractions. Protein concentration measured by spectrophotometry, reading absorbance at 280 nanometers, aromatic amino acids, tryptophan, and tyrosine absorbing UV light proportional to protein concentration. Purity assessed by SDSPA, GE electrophoresis, protein samples mixed with reducing agent-breaking disulfide bonds, heating to denature proteins, loading onto polyacrylamide gel, running electrophoresis separating proteins by molecular weight, staining gel with Kumasi blue dye, revealing protein bands. Single band at approximately 160 kilodaltons indicates pure Cas9 without contaminating proteins. Synthetic guide RNA ordered from commercial suppliers specializing in oligonucleotide synthesis or transcribed in vitro using T7-RNA polymerase enzyme, synthesizing RNA from DNA template containing T7 promoter sequence followed by desired guide RNA sequence. In vitro, transcription reaction contains DNA template, T7 polymerase, 4 ribonucleoside triphosphates, ATP, CTP, GTP, UTP, serving as building blocks, buffer providing optimal pH and magnesium concentration. Incubating at 37 degrees for several hours allows polymerase transcribing thousands of RNA copies per DNA template. Template DNA degraded by adding DNA-SE enzyme, specifically cleaving DNA, leaving RNA intact. RNA purified by phenol chloroform, extraction removing proteins, followed by ethanol precipitation concentrating RNA, washing with 70% ethanol removing salts, air drying pellet, resuspending in nucleus-free water. RNA quantified by spectrophotometry, measuring absorbance at 260 nanometers, Quality checked by denaturing gel electrophoresis, ensuring single band at expected size without degradation products. Target DNA substrate prepared, creating defined test sequence for examining cleavage specificity. Short DNA segments amplified by PCR using primers designed to generate fragments several hundred base pairs, long containing target sequence complementary to guide RNA spacer sequence. PCR products purified using spin columns binding DNA to silica membrane in presence of chaotropic salts, washing away primers and DNTPs and buffer salts, eluting in water. Alternatively, longer substrates prepared by digesting plasmid DNA with restriction enzymes cutting at specific recognition sequences, generating linear DNA fragments isolated by gel extraction cutting desired band from agarose gel after electrophoresis, purifying DNA from gel slice. Cleavage assay. Experimental setup requires careful attention to reaction conditions. Cas9 protein and guide RNA premixed in binding buffer, incubating at room temperature, allowing ribonucleoprotein complex forming. Cas9 conformational changes occurring upon guide RNA binding, activating nuclease domains. Target DNA added initiating cleavage reaction, magnesium chloride required as cofactor for nuclease activity, buffer maintaining physiological pH and salt concentration, total reaction volume typically 20 microliters, conserving expensive purified components. Reactions incubated at physiological temperature, typically 37 degrees Celsius, for 30 minutes to 1 hour, depending on enzyme concentration and substrate accessibility, allowing Cas9 guide RN1A complex scanning DNM-A, searching for protospacer adjacent motif, PAM sequence NG, G required for Cas9 binding. When PAM found, checking whether adjacent sequence matches, guide RNA spacer. If matching Cas9, inducing local DNA melting, allowing guide RNA base pairing with target strand, activating nuclease domains cutting both DNA strands, approximately 3 base pairs upstream of PAM sequence. After incubation, reactions stopped by adding EDTA, chelating magnesium, analyzed by agarose gel electrophoresis. Successful cleavage produces 2 DNA fragments corresponding to cut sites. Unsuccessful reactions show uncut full-length DNA. You watch researcher loading gel, running electrophoresis, staining with ethidium bromide, imaging under UV light. Results clear. Guide RNA specifically directs Cas9 cutting predicted target site, leaving unrelated DNA uncut, demonstrating programmable sequence-specific DNA cleavage. Paper published June 2012 in Science Journal, demonstrating CRISPR-Cas9 as programmable RNA-guided DNA endonucleus, immediately recognized as transformative technology. Within months, multiple laboratories demonstrate CRISPR working in mammalian cells, editing genes in human cell lines, mice, plants, essentially every organism tested. Technology spreads rapidly through research community, thousands of laboratories adopting CRISPR for genetic experiments, commercial suppliers offering custom guide RNA synthesis, detailed protocols shared freely online, democratizing gene editing, similar to how PCR democratized DNA amplification 3 decades earlier. CRISPR's simplicity and versatility drive explosive adoption. Previous gene editing technologies required extensive protein engineering for each target, limiting accessibility to specialized laboratories with expertise. CRISPR requires only designing 20 nucleotide guide RNA sequence complementary to target, synthesized quickly and inexpensively by commercial suppliers, dramatically lowering barriers to gene editing. Applications multiply, creating cellular disease models by introducing mutations, generating knockout mice lacking specific genes, engineering crop plants with improved traits, studying gene function systematically, correcting disease-causing mutations in patient cells for therapeutic purposes. You watch laboratory transformed by CRISPR's arrival. Previously challenging experiments become routine. Graduate student designs guide RNAs computationally using online tools. Orders synthetic guides arriving within days. Transfects guides plus Cas9 into cultured cells using standard transfection reagents. Selects edited clones using antibiotic resistance or fluorescent markers. Validates editing by sequencing target locus, confirming intended modifications. Process takes weeks rather than months or years required by previous methods. Experimental velocity accelerates dramatically, questions previously considered too difficult becoming tractable, systematic screens testing hundreds of genes becoming feasible using pooled guide RNA libraries. But therapeutic applications require additional considerations beyond cellular experiments. Delivering CRISPR components into patients' bodies challenges Cas9, protein, large and RNA-guided complex requires both protein and RNA entering cells, immune responses against bacterial Cas9 protein, potentially limiting repeated treatments, off-target effects where CRISPR cuts unintended similar sequences causing unwanted mutations must be minimized. Different delivery strategies developed, viral vectors carrying Cas9 and guide RNA genes, lipid nanoparticles encapsulating CRISPR components, direct protein RNA complex delivery, each approach having advantages and limitations depending on target tissue and application. You drift forward, finding yourself at Children's Hospital of Philadelphia, year 2017, observing first FDA-approved gene therapy, clinical trial using Lux Turner, treating inherited retinal dystrophy caused by mutations in RPE65 gene. Treatment involves injecting adeno-associated virus vectors carrying functional RPE65 gene directly into retina beneath photoreceptor cells. Viral vectors transduce retinal pigment epithelium cells, delivering working gene copy, allowing cells producing functional protein, restoring visual cycle in photoreceptors, preventing progressive blindness, even restoring some vision in patients already experiencing significant vision loss. You observe clinical procedure. Patient under anesthesia. Ophthalmologic surgeon performing vitrectomy, removing vitreous fluid from eye creating space for injection, inserting fine needle through sclera into subretinal space between retinal pigment, epithelium and photoreceptors, slowly injecting suspension containing billions of viral vector particles, creating temporary retinal detachment as fluid lifts retina, allowing vector contacting target cells. Procedure delicate, requiring steady hands and precise technique, performed under surgical microscope magnifying view. After injection complete, retina gradually reattaches over days as injected fluid absorbed. Viral vectors transduce cells, transgene expression begins within weeks. Protein production continues for years from episomal vector DNA persisting in non-dividing retinal cells. Patients' vision improvements documented through visual acuity testing, light sensitivity measurements, mobility testing in dim light obstacle courses. Many patients experience significant improvements, legally blind individuals gaining functional vision, enabling reading, recognizing faces, navigating independently. Treatment represents milestone, demonstrating gene therapy's therapeutic potential for monogenic diseases where single gene defect causes condition, functional gene. Copy can correct problem, accessible tissue allows direct vector delivery. FDA approval. October 2017 marks gene therapy's arrival as mainstream medical treatment after decades of research and early setbacks. Additional gene therapies follow. Zolgensma approved May 2019 treats spinal muscular atrophy, fatal genetic disease where mutations in SMN1 gene cause motor neuron degeneration, progressive muscle weakness, death in infancy. Treatment delivers functional SMN1 gene using adeno-associated virus vector administered intravenously, transducing motor neurons throughout spinal cord, halting disease progression, allowing affected infants achieving motor milestones previously impossible, some learning walk independently, despite devastating prognosis without treatment. Single infusion costing over $2 million, most expensive drug ever approved, yet potentially curative treatment, obviating lifetime of medical interventions and dramatically improving quality of life, justifying extraordinary cost. You stand in hospital room watching infant receiving Zolgensma infusion. Baby lying in crib with monitoring equipment tracking vital signs, intravenous line in tiny arm or leg, delivering vector slowly over approximately 1 hour. Parents sit nearby, anxiously hopeful, knowing treatment offers chance their child surviving and thriving rather than succumbing to disease within months. Medical team monitors closely for adverse reactions, most patients tolerating treatment, well though some experience liver enzyme elevations requiring monitoring and management. Treatment timing critical. Earlier treatment before extensive motor neuron loss provides better outcomes. Newborn screening programs increasingly identifying affected infants, enabling treatment before symptoms appear, maximizing benefit. These approved gene therapies treat monogenic diseases caused by single gene mutations, relatively straightforward conceptually, though technically challenging, requiring years of preclinical development, clinical trials, regulatory review. But most common diseases involve complex genetics, multiple genes interacting with environmental factors, presenting greater therapeutic challenges. Cancer, particularly promising target, CAR T-cell therapy, genetically modifying patients' own T cells to recognize and attack cancer cells, showing remarkable responses in otherwise treatment-resistant blood cancers. Ex vivo gene editing using CRISPR enables creating enhanced CAR T cells with additional modifications improving persistence and efficacy, beginning human trials for multiple cancer types. You drift toward future-watching emerging technologies building upon CRISPR foundation. Base editing developed 2016 by David Liu Laboratory chemically converts single DNA bases without cutting DNA, fusing catalytically inactive Cas9 to deaminase enzymes converting cytosine to uracil or adenine to inosine, creating permanent C to T or A to G changes, enabling correcting point mutations without double-strand breaks, potentially causing unwanted insertions, deletions, or chromosomal rearrangements. Prime editing developed 2019 achieves all 12 possible base conversions plus small insertions and deletions using Cas9 fused to reverse transcriptase. Copying desired edit from prime editing guide RNA directly into genomic DNA, writing any sequence change without requiring DNA templates or double-strand breaks, greatly expanding editing precision and versatility. Epigenetic editing enables controlling gene expression without changing DNA sequence, targeting CRISPR to regulatory elements, recruiting activator or repressor domains, turning genes on or off reversibly, useful for conditions where gene activity needs modulation rather than permanent sequence change, potentially treating conditions caused by inappropriate gene silencing or overexpression. Multiplexed editing allows simultaneously modifying many genes using multiple guide RNAs, enabling complex genetic rewiring, constructing synthetic gene circuits, wholesale genetic programming, approaching science fiction capabilities. Germline editing represents controversial frontier, modifying sperm, eggs, or embryos, creating heritable changes passing to future generations. He Jianqui's unauthorized 2018 experiments editing human embryos and implanting them, producing CRISPR-edited twins with modified CCR5 gene, supposedly conferring HIV resistance, sparked international outcry over ethics of heritable human genetic modification, lack of medical necessity, inadequate consent procedures, insufficient safety data, need for international governance. Subsequent international consensus recommends moratorium on clinical applications of germline editing until safety, efficacy, and ethical considerations adequately addressed. Though basic research on embryos continues understanding development and evaluating editing approaches. You find yourself in laboratory equipped for synthetic biology, field engineering biological systems using engineering principles, designing genetic circuits performing logic functions, building biosensors detecting environmental signals, creating metabolic pathways, producing valuable compounds, programming cells as living factories. CRISPR accelerates synthetic biology dramatically through enabling rapid construction of strains with multiple genetic modifications, testing design iterations quickly, building increasingly complex engineered systems. Microorganisms engineered, producing biofuels from renewable feedstocks, synthesizing pharmaceutical precursors traditionally extracted from plants, manufacturing industrial chemicals through fermentation rather than petroleum-based chemistry, potentially transforming chemical industry towards sustainable biological production. Genome synthesis technologies enable writing entire chromosomes or genomes from scratch. DNA synthesis costs dropping exponentially, similar to sequencing costs, enabling creating designer genomes optimized for specific applications. Craig Venter's synthetic genomics team created first synthetic bacterial genome 2010, synthesizing entire mycoplasma, mycoids chromosome chemically, transplanting into different bacterial species, successfully booting up synthetic genome, creating functional organism with DNA designed and constructed in laboratory. Subsequent efforts aim creating minimal genomes containing only essential genes required for life, understanding fundamental principles of cellular function, creating chassis organisms for synthetic biology applications with streamlined genomes optimized for bioengineering. Laboratory atmosphere reflects synthetic biology's interdisciplinary nature, biologists working alongside engineers, computer scientists, physicists, approaching biology as engineering discipline, using standardized biological parts, modular genetic elements, computational design tools, automated construction platforms, high-throughput testing systems evaluating designs. Equipment includes liquid handling robots constructing DNA assemblies, automated cell culture systems, flow cytometers analyzing fluorescent reporters indicating circuit behavior, fermentation vessels producing engineered microorganisms at scale, whiteboards covered with genetic circuit diagrams resembling electronic schematics, promoters acting as switches, genes as functional modules, regulatory elements as control systems, biological systems conceptualized using engineering abstractions. You watch research team debugging genetic circuit not functioning as designed. Troubleshooting process, familiar to software engineers or electrical engineers, identifying component not working correctly, replacing or modifying element, retesting, iterating until circuit performs desired function. Genetic circuit design databases collect characterized parts with quantified properties, enabling predictive design using mathematical models, though biological complexity means models imperfect. Experimentation still essential for validating designs. Biology's messiness, resisting complete reduction to engineering principles, remaining fundamentally discovery science despite engineering approaches. Future possibilities seem simultaneously exciting and sobering. Genetic technologies offer unprecedented power, preventing and treating diseases, enhancing agricultural productivity, creating sustainable industrial processes, understanding life's fundamental mechanisms. Yet same technologies raise concerns about unintended consequences, equitable access, potential misuse, appropriate governance. Designer babies, genetic enhancement, extinct species, resurrection, creation of novel organisms, gain-of-function research on pathogens, bioweapons potential, each application raising ethical questions requiring societal dialogue beyond scientific considerations alone. You stand in quiet genomics laboratory late evening, after regular hours when most staff departed for home. Automated DNA sequencers continue running, LED indicators blinking, capillaries separating fluorescent fragments, data accumulating. PCR machines complete final thermal cycles, samples cooling to storage temperature. Cell culture incubators maintain precise 37-degree environment. Genetically modified cells growing steadily doubling every day. Liquid nitrogen freezer hisses softly, maintaining minus 180-degree temperature, preserving cell lines and valuable samples. Computers process data continuously, sequence alignments updating, gene expression analyses computing, assembly algorithms building contigs. Single researcher remains working late, sitting at computer analyzing CRISPR, editing results from today's experiments. Screen shows DNA sequencing chromatograms, comparing edited sequences against reference, quantifying editing efficiency, identifying any unexpected modifications at off-target sites. Most edits successful, few showing minor indels at target sites, no significant off-target mutations detected. Experiment succeeded according to plan. Researcher makes notes in electronic laboratory notebook, saves analysis files to network drive, considers next experiments extending findings, testing additional guide RNAs, evaluating different cell types, optimizing protocol parameters. Laboratory quiet except for equipment background sounds, refrigerator compressor cycling, biosafety cabinet fan humming, occasional thermal cycler beep indicating completed run, familiar nighttime laboratory soundscape. Researcher stretches, yawns, checks time on computer, nearly midnight, good stopping point. Tomorrow will run confirmatory experiments, repeat successful conditions verifying reproducibility, prepare figures for presentation at lab meeting next week. Powers down computer, turns off most lights leaving one for safety. Checks incubator temperatures one last time, ensuring cell cultures maintaining proper conditions overnight. Arms security system, locks door, walks through empty hallway. Outside research building, night air cool and refreshing after hours in climate-controlled laboratory. Campus quiet, few people walking, streetlights casting pools, illumination along pathways. Researcher walks toward parking lot thinking about experiments, mentally planning tomorrow's work, satisfied with progress, but aware how much remains unknown, each question answered raising new questions, biology's complexity perpetually unfolding, revealing deeper layers, understanding. Genetic journey continues, from Mendel's garden peas, counted patiently across seasons, through Morgan's fly-room mutations, Avery's transformation principle, Watson-Crick's elegant double helix, Meselson-Stahl's replication proof, genetic code decipherment, Sanger's sequencing, PCR's amplification, Genome Project's completion, CRISPR's editing precision. Each breakthrough building upon previous discoveries, cumulative progress, advancing understanding incrementally through careful experimentation, thoughtful interpretation, technological innovation enabling previously impossible observations. Future holds promise of personalized medicine, matching treatments to individual genetic makeup. Genetic diseases prevented through early intervention or corrected through gene therapy. Agricultural improvements feeding growing populations sustainably. Biological manufacturing replacing petroleum-based chemistry. Understanding life's fundamental principles at molecular level, enabling previously unimaginable applications. Yet with power comes responsibility. Genetic technologies requiring thoughtful governance, ensuring benefits distributed equitably, risks managed appropriately, ethical considerations guiding applications, societal dialogue shaping how technology is deployed, balancing innovation's pace with wisdom's caution, optimism tempered by humility. Recognizing biology's complexity exceeds current understanding, unintended consequences potentially arising from even well-intentioned interventions. Science continues patient work, asking questions, designing experiments, analyzing results, publishing findings, debating interpretations, refining understanding through iterative process, extending across generations of scientists, building collectively upon shared knowledge. Patient machinery of scientific progress, continuing day by day, experiment by experiment, discovery by discovery. Day by day. You sit now in empty laboratory after hours, night deepening outside windows, equipment humming softly maintaining constant operation. DNA continuing quiet work throughout building and across world, copying itself through patient semi-conservative replication, transcribing genes into messenger RNA intermediates, translating RNA sequences into amino acid chains, folding into functional proteins. Being edited with molecular scissors at programmed sites, analyzed through sequencing, revealing information encoded in chemical letters, understood progressively through patient observation and careful experimentation accumulated across generations. Somewhere in building cells grow steadily in temperature-controlled incubators. Bacterial colonies form overnight on agar plates. PCR machines amplify specific DNA targets through repetitive thermal cycling. Automated sequencers call bases from fluorescent signals. CRISPR systems cut precisely at sites determined by 20 nucleotide guide sequences, genetic information flowing through patient machinery of molecular biology, continuing through night's welcoming silence. Rest now peacefully. Sleep among nucleotides and laboratory benches. Patient genetic work continues always without pause. DNA quietly performing its elegant molecular duties, copying itself faithfully, maintaining information across generations, encoding instructions precisely through simple four-letter alphabet. Subject to patient investigation, revealing nature's solutions to biological information storage and inheritance problems solved through evolutionary time.